And now I think everybody can hear me. All right. I'm just testing things, but okay. Preset number 3 doesn't really do anything. It is. Yep. I just feel like this is a little too. This is a little bit better, I think. Is it okay? Like, now it looks like we're in a planetarium. Huh? It's either we're gonna try. I don't know what the different presets do. We're gonna have to go full. We're gonna have to go all on. That's this room, it works that way. So anyway, hello. It's 6 o'clock, so let's get started. Um, those of you who for those of you who are not in the physiology course, um, my name is Dr. Patricio Mujica, and I will have the distinct pleasure of making your life a little bit more difficult the next 2 weeks. So today we have to cover three related topics. In the first hour, we're going to talk about the nucleus, then we're going to talk about the cell cycle, and then at the end, we're going to discuss apoptosis. Perfect. Now this is much better. So let's just get started. Dr. Breidenreich, do you have anything you need to say? Excellent. Okay, let's get going. So, and there, yeah, fine. So we are recording, everything's okay. But I need to click here. Perfect. Okay, learning objectives for today. I'm not going to read them because, that would be boring. But you can do that on your own. Again, this is something that you're going to see in the first set of slides. Each slide covers a particular topic. Sometimes some topics are covered by more than one slide, but at the bottom you'll see that the specific learning objective that content is associated to. So you can hopefully use that as to guide your study. This always happens. I apologize, but these timings are the death of me. So this is something that I just did, in the past half hour. So I don't have them for these two, for the second and the third hour, but you'll be able to figure it out. And let's prevent my family from bothering me. All right, so we're going to start with basically just something very simple, which is the nuclear structure, right? Eukaryotic cells that actually do have a nucleus, basically have the ability to contain most of their genetic material within this compartment that is characterized by the elements that you can see here. On the left, this is an electron micrograph of the nucleus of a particular cell. On the right, what you have is a sort of a cartoon that represents most of the important structures that serve to give the nucleus its particular structure, right? So the nucleus is bounded by a double membrane that's called the nuclear envelope. The nuclear envelope is basically an extension of the cell's endoplasmic reticulum. And the reason for this is probably probably evolutionary, in the sense that early eukaryotic cells probably just started developing additional membrane in order to basically in order to support their growth. And that led to, oops, this is, the gain here is a little high. This is, there we go. So and now they can't hear me probably. So, hello. Apologies for the technical problems. Anyway, there's something in the chat. Am I? Okay, you can hear me. Thank you. I appreciate it. So again, the nuclear envelope developing evolutionarily from the just growth of the cell and the necessity to basically just make more membrane that led to the development of the endomembrane system of the cell that basically ended up compressing the genetic material of the cell. Inside the nucleus, on a cell that's not dividing, the DNA, the genetic material, is found in the state of chromatin. So basically, this is much less condensed form or structure of DNA. And we're going to talk quite a bit about that in the next few minutes. And chromatin can usually be found in two states: heterochromatin, which is extra condensed, and that refers to those regions there that are sort of in slightly more, slightly darker blue. And then euchromatin, which is a little more relaxed, and it tends to be associated with regions of the DNA that are undergoing active gene expression. The DNA itself is also associated with, the nucleus is usually has a structure that's called a nucleolus. Many cells have more than one nucleolus. Other cells have just one. And it represents a region or a condensate of specific regions of DNA that are actively making ribosomal RNA. So there's something happens there, and the details of how and why are beyond the scope of the course. But this is important to realize that many nuclei have these nucleolus or nucleoli, dependent, right? And something that's important, we're going to talk a little bit about that later on, is the presence of a nuclear lamina. So it's represented by this band in brown here. It's a network of structural proteins that basically keep the shape of the nuclear envelope in place. And it basically helps maintain the structural integrity of the nucleus. Lamins are also important so regulation of lamin integrity is relevant in the ability of the cells to time the progression of the cell cycle. So we're going to say a couple of things about them later on. Peripheral to the nucleus, so outside, usually we find centrosomes or microtubule organizing centers that are important in the transition from a quiescent cell to a dividing cell. And that's basically it. The last thing that I didn't mention before is the presence of the nuclear pores, right? The nuclear pores, we are going to mention them in detail later on. The nuclear pores enable the transport of materials, the exchange of materials, proteins and RNA, DNA, whatever, between the nucleus, the nucleoplasm, the contents of the nucleus, and the cytosol of the cell. So that's also an important detail. So this should be clicking, but it's not clicking. And now it clicks. So let's start with talking about the DNA, the ways that the DNA is structured in the nucleus of the cell. So researchers in the lab can isolate the DNA from non-dividing cells, so DNA that you would normally expect to be in the state of chromatin, and you can put this DNA in a little, say, glass support, and then you can basically kind of extend it and take a picture of it under the electron microscope. So this figure shows these two states of condensation for chromatin. So on top you have isolated chromatin that's directly isolated from the nucleus, and then you can sort of pull it to reveal the structure of the string or decondensed chromatin, where you can actually observe those little beads, that sort of beads in a string type of architecture that characterizes chromatin structure. Now, what you see here, this is a picture of actual real chromatin. The structure that makes it the way it is, that makes it to be the way it is basically this conformation that people call it beads on a string, and it consists of nucleosomes, which is basically a core of proteins surrounded by 2.5 or so turns of the DNA double helix molecule, and then a string of linker DNA that you can see in between these nucleosomes, right? So the nucleus, the length of the DNA molecule that surrounds an entire core plus a linker is about 200 nucleotides. There are about 150, I think the actual number is 147 nucleotides that are wrapped around the core, the nucleosomal core, the nucleosomal protein core. You can take this beads on a string, and then you can treat it with a nuclease, so an enzyme that degrades DNA, and you can isolate the nucleosome particles, and you can basically further treat that with high concentrations of salt to dissociate the DNA from the protein core, and you can basically study them independently of each other. So the core of the nucleosome is made of 8 proteins called histones. We sometimes call that the histone octamer. And then again, 146, not 147, 146 nucleotide pairs of nucleotides. So basically 146 pairs of nucleotides, I suppose. And you can further dissociate these things, and you can see that the octamer basically is formed of 4 pairs of histones. There is histone H2A, there are 2 of those, 2 of H2B, 2 of H3, and then 2 of H4. Those are the 4 histones that make up the core octamer of the nucleosomes, right? You might wonder, where is H1, right? Is there an H1? And the answer is yes, there is an H1, but we are going to talk about it in just a bit. So in that bit has come to us. The H1 histone is actually a molecule that is found in between the actual nucleosomes. It serves a really important purpose in linking adjacent nucleosomes, and that's why it's called the linker histone. So H1 basically binds to the DNA, and it helps pack these nucleosomes and stack them in a structure that further condenses the structure of chromatin, right? So again, H1 is important in the compaction, in the further compaction of chromatin structure. And what can we do with this compaction? So this figure basically shows you the different levels of organization of eukaryotic DNA. So you start with the actual double helix, which has basically a width of about 2 nanometers. And this double helix wraps around the octamers of histones and creates the beads and strings structure that is about 11 nanometers in width. That's why it's called the 11 nanometer fiber. The beads in a string structure can actually, using with the help of the H1 histone, can compact itself into the 30 nanometer fiber that is further coiled, and the nucleosomes are basically packed on top of each other, sort of pancaked. They stack on top of each other. And the 30 nanometer fiber can then further condense into the 300 nanometer fiber. And the 300 nanometer fiber can further coil and further condense itself to create basically the width of an arm of the chromosome, right? So when the DNA is fully compacted, what you get is basically the actual chromosome, which is about 1400 nanometers in width, and it contains a ridiculous amount of DNA. So there's these molecules are really, 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 really long. So the net result is that each DNA molecule can be packaged into a chromosome, into what we call a mitotic chromosome, and it's a condensation that's 10,000 fold, right? So in other words, the length of the entire chromosome is 10,000 fold shorter and more compacted than the actual length of the naked DNA molecule. Something that's I've always found it to be an interesting factoid is that each chromosome is a single DNA molecule, right? So if someone ever asks, how could you know how many DNA molecules a human cell has? The answer is 46. We have 46 molecules of DNA in each one of our cells, right? Each molecule forms one chromosome. Okay, so there are certain irregularities, right? So the 30 nanometer fiber, so this particular structure in the chromosome, in the chromatin molecule, doesn't only contain DNA and histone proteins. There are tons of non-histone proteins. And there's good reason for that, right? So these non-histone proteins actually are the ones that are involved in, they help compact the chromosome, they help structure the chromosome, but they also are involved in the regulation of gene expression. So the idea is that at any given time, the total mass of histones is equal to the total mass, or roughly equal to the total mass of non-histone proteins. And these non-histone proteins, again, the nature, the functions of these non-histone proteins is very, very, very variable. But at this point, all you need to know is that these proteins exist. And, based on what you know, or hopefully will leave this session knowing, you'll have an idea of what kind of things these non-histone proteins do. In total, a particular chromosome is about 1/3 DNA and 2/3 protein by mass. So if you were able to just take one chromosome and basically weigh it or estimate its mass, 1/3 of that mass is going to be DNA, 2/3 is going to be protein. Now, to talk a little bit more about the structure of histones. Core histones have this an N-terminal tail. So on the amino terminus of the amino acid sequence, this is perhaps the most significant region of the histone in terms of its ability to be regulated. So most of the regulation that happens to histones happens in this N-terminal domain or the N-terminal tail. And for a number of reasons, this N-terminal tail is called a tail because it actually protrudes outside of the nucleosome. I'm going to show you a couple of, I'm going to show you a figure that sort of depicts that. Give me a sec. There we go. So besides the N-terminal tail, they have this histone fold, which basically forms the actual core of the histone. So those are the parts that basically fold in such a way that form this nucleus, this core that DNA sort of wraps itself around, right? So these are, generally speaking, the two important things about the structure of histones. Their N-terminal tail and the histone fold. The fold itself is sort of, it operates kind of as a handshake. So basically, the histone fold contains three general domains, and they fold in such a way that the shorter domains are interacting with the ends of the central domain, and the thing here that's important to realize is that there are pairs of histones that basically have kind of a handshake between them. So H2A and H2B interact in this particular way. Again, it's like they're shaking their hands. H3 and H4 do something similar. And when we put all of these things together, what this is basically, this figure shows kind of how all of the elements that we are discussing sort of interact. This pinkish, I don't know what color you see, but this pinkish band of sorts represents the molecule of DNA. And at the center, you have the histone core, two molecules of H2, two molecules of H2A, two H2B, two H3, two H4. And the histone fold is at the center. The histone tails are protruding to the outside, right? This is important in the sense that because these histone tails are facing and sort of peeking outside of the nucleosome, outside of the core of the nucleosome, they can be seen by enzymes that regulate their activity. And we're going to discuss a little bit more on that, right? So first, we're going to say a couple of words about the way that the histones interact with the DNA itself. The idea is that the molecule of DNA, you know, that the molecule of DNA has a what we call a minor groove and a major groove. So generally speaking, the sections of the DNA molecule that directly interact with the histone, they tend to be rich in dinucleotides that are either diadenines, dithymidines, or a mixture of both. So AA, TT, or TA directly interacting with the nucleosome core, and guanines and cytosines facing outwardly, right? And this is this is made possible in all likelihood by the non-covalent interactions that that occur between the DNA molecule and the nucleosome. All right. So no, why?
No, okay, I don't know, you just pause.
I paused because I was reading something on the chat.
Oh.
So there we go. Also setting a couple of things up here is weird. Anyway, so what happens to the histones, right? How do we basically help the histones do whatever it is that they need to do? The histones, again, as I said before, they have their tails, and the tails of the histones are exposed to the sort of the outside of the chromatin fiber, and therefore they are accessible to enzymes that can covalently modify them. When we say covalent modifications, we're talking about phosphorylation, but there are a couple of other types of covalent modifications that are relevant here that are perhaps better studied in histones than in other types of proteins. So first of all, there are interactions between the tails that can help pack the higher order structures of the chromatin. And that is made possible by the fact that the histone tails have a relatively unstructured structure. That's sort of an oxymoron, but basically they don't have like constraining secondary structures, like the way that alpha helices or beta pleated sheets are or can be constricting. These are more free amino acid sequences. So that's what we mean by when we say that they are unstructured and highly mobile. So on this figure to the right, what you have are the four main core histones, so H2A, H2B, H3, and H4. These are the amino acid sequences of their N-terminal tails, right? And highlighted in each one of them are the most common forms of post-translational modifications that happen in each. So basically first of all, the thing that you need to pay attention to here is there are basically three types of modification that happen in histones: methylations, phosphorylations, and acetylations. Phosphorylations you're probably the most familiar with. Methylation and acetylation are similar. They work in a similar fashion. They basically there are enzymes that add either an acetyl group or a methyl group, the same way that kinases add phosphate groups. But what's different here is the residues on which this happens. Phosphorylations tend to happen either on serines, threonines, or tyrosines, right? And that's, you probably know the amino acid one letter code. So the S is a serine. Most of the phosphorylation sites here are serines. Acetylations and methylations tend to happen on arginines or lysines, right? Arginines or lysines. And arginine is the R, lysine is the K. And what do those two amino acids have in common, lysines and arginines? They're basic, they're positively charged, right? So now think of, this is one way of thinking about how these things basically operate in the end. Positive residues in proteins may interact with negative groups in the DNA molecule. The DNA molecule is negatively charged. So when we modify these positively charged amino acids like lysine and arginine, what we end up doing is basically changing the ability of those residues to interact with other charged particles, right? And that either creates that may increase the likelihood of interaction, or it may decrease the likelihood of interaction. The details of that and how all of that comes together is called the histone code. There are combinations of these post-translational modifications that are associated with increased gene expression, others that are associated with decreased gene expression. So the way that they work is very complicated, and we're not really going to go into the details of it. What you do need to know is the types of modifications, the amino acids where they happen, not the details of, you don't need to memorize, there's an acetylation of lysine 5 in H2A. You need to know that lysines can acetylate it or methylate it, or both. Actually, there are several, for instance, lysine 9 in H3 can be either methylated or acetylated. And phosphorylation happens in serines primarily, right? So I was talking about the histone code. Combinations of these post-translational modifications do have an important role in regulating gene expression. So for instance, we're going to take the case of H3 and all of these different possibilities. You can methylate arginine 2, lysine 4, lysine 9, blah, blah, blah. All of those things can happen. And they may happen simultaneously. However, there are specific signatures that are associated with specific outcomes in gene expression. So, for instance, the trimethylation of lysine 9 in H3 is associated with the formation of heterochromatin, and heterochromatin is associated with the silencing of gene expression, right? Remember, heterochromatin is a state that's more compacted, and therefore it prevents access to the enzymes that mediate, transcription or transcription factors, right? On the other end, trimethylation of lysine 4 and acetylation of lysine 9 are associated with the opposite, with gene expression. So what this combination of modification causes is basically for the nucleosomes to be less packed together so that the molecule of DNA can actually be exposed to transcription factors, to transcription enzymes, and all of these mechanisms that and enzymes that mediate gene expression. Johan in the chat is asking whether you can say that acetylation and methylation happens in positive amino acids. What we're going to say is that they happen in lysines and arginines, because to the best of my knowledge, histidine, also a positive amino acid, is not associated with this, right? So don't make that generalization. Just focus on the specific amino acids that are involved. Lysine and arginine. Okay, so far, any questions? Anything in the chat? I don't think so. Okay, so the nuclear envelope. As I said before, the nuclear envelope is a double membrane, right? So it's not just a lipid bilayer, it's a lipid bilayer that sort of folds on itself, and it creates this double layer. And the space between the two layers of the nuclear envelope is continuous with the lumen of the endoplasmic reticulum, right? It's continuous with the lumen of the endoplasmic reticulum. So basically, this cartoon shows sort of the way that the nuclear envelope is structured. This white space in between in between, that's the lumen of the ER here. It's the space in between the or what we call the perinuclear space. The inner leaflet of the nuclear envelope contains the nuclear lamina, which is this basically it's a meshwork of structural proteins similar to akin to the cytoskeleton in the cytosol, that basically keeps the structure of the nuclear envelope together. And the nuclear envelope itself is perforated by these nuclear pores, and the nuclear pores contain nuclear pore complexes. So these are very complicated groups of proteins that are lining those pores, and as we're going to see a little later, they mediate the they not only mediate, but they regulate the traffic of material between the nucleus and the cytoplasm of the cell. Speaking of the nuclear pores and the nuclear pore complexes, they have very complicated structures. They contain a ring of membrane proteins that basically anchor these scaffold proteins in red and yellow. And these proteins in the proteins in red and yellow are called nucleoporins. And there are scaffolding nucleoporins that basically anchor everything else to they help anchoring everything to the membrane. And then the yellow ones are called channel nucleoporins that basically create this fibrous meshwork that serves a little bit as a filter through the channel of the pore. And on top of that, there are proteins that form this basket that you see on the nuclear side, right? So the cartoon basically is a representation of the structures that are seen in these other parts of the figure here. This is actual data from these are electron micrographs of nuclear pores and nuclear pore complexes. The thing that I didn't mention before are the importance of these cytosolic fibrils. So these are sort of projections of these scaffold nucleoporins that, again, they project towards into the cytosol, and they function a little bit as antennae. And I want you to remember that because that's important in the function of some of the regulators that control the movement of stuff through the pore. The nucleoporins there's many different kinds. There are about 30 different types of proteins making the nuclear pore complex, and many of them are present in multiple copies. And they are arranged symmetrically, so you can basically see from this figure that there is a certain symmetry to the organization of the pore. And we can classify them in multiple different ways. There are the transmembrane ring proteins that are the green ones. And again, they anchor the envelope to they anchor the whole complex to the actual envelope. There are the scaffold nucleoporins that I was mentioning before. They form these are the red ones in the figure. There are the channel ones that line the pore. They control the flux of things through the nuclear pore. And then there are these fibrils that I was mentioning a little earlier. So the cytosolic fibrils facing the nuclear side, so basically they converge and they form this basket that is important for nuclear pore function. And then there's this meshwork of untangled, disordered protein segments that functions a little bit like a filter, right? And they block the passive of large macromolecules, right? They serve a little bit as a filter, right? Now, how do these things work? The nucleoporins form this filter system that excludes molecule based on size. Each nuclear pore complex can transport up to 1,000 macromolecules per second. So this is a relatively fast rate of transport. And this figure here, it's a tad older, but it still represents the same structures, right? So there you have the scaffolding. Here you have again the scaffolding nucleoporins, then the channel nucleoporins, and these gray things represent the disordered fragments. The transport of material between the nucleus and the cytosol is simultaneous. There's no actual there's no intrinsic directionality to the transport. So things can move in both directions, from the nucleus to the cytosol, or from the cytosol to the nucleus at the same time using the same pore. Something that characterizes the traffic of materials between the cytosol and the nucleus is that smaller proteins and smaller macromolecules can actually permeate pretty much freely, right? So they can kind of diffuse between the cytosol and the nucleus, so long as they are less than 5 kilodaltons in size, right? So that's your cutoff. If you're larger than 5 kilodaltons, then you cannot. The filter function of the nuclear pore will prevent that from happening. Larger stuff will diffuse much more slowly, right? And again, the larger the molecule, the slower it's going to diffuse. And things that are things that are larger than 60 kilodaltons cannot enter by passive diffusion, plain and simple, right? So if you are smaller than 5 kilodaltons, you can move freely between the nucleus and the cytosol. If you are between 5 and 60, the rate at which that diffusion will happen is going to be proportional to the size of the molecule, right? The larger it is, the slower it's going to diffuse. But above 60 kilodaltons, you cannot, period, diffuse between the nucleus and the cytosol. All right. Now, the thing is that there is quite a bit of nuclear components that are synthesized by cytosolic ribosomes. So how do we make sure that the things that need to go into the nucleus actually make it into the nucleus in a timely manner? So we have to have a mechanism that ensures that everything that needs to be in the nucleus, regardless of its size, and particularly important, this is particularly important for the larger stuff, we have to have a mechanism that ensures that that material makes it into the nucleus. And in eukaryotic cells, this mechanism is based on nuclear localization signals, right? There's basically a string or a sequence, not a string, but a sequence of amino acids that has been determined to be a nuclear localization signal. So in this case, we have our cells that have been infected with a virus, the SV40 virus. And the SV40 virus delivers a particular antigen that's called the T antigen. And that T antigen basically needs to make it into the nucleus in order to do the things that it needs to do for the viral infection. On the left side, what you have are normal viruses that contain the normal nuclear localization signal on their T antigen. And that signal is lysine, 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 arginine, lysine. So again, sort of a string of positively charged amino acids. When the investigators took the DNA molecule the DNA sequence that encodes for this antigen and mutated it so that one of those, the second lysine here, is mutated to a threonine, which is it's a neutral amino acid. And then they infected the cells with this mutated virus that basically has a messed up nuclear localization signal on this particular protein. The protein was prevented from going into the nucleus. So you see here, the protein is located everywhere in the cell except the nucleus, which the nucleus is a little dark, right? Compare that to this image on the left, where the protein is only in the nucleus, right? This is a very good demonstration that the nuclear localization signal is necessary for the localization of proteins to the nucleus. Now, the proteins have these nuclear localization signals, but these signals actually have to operate in a particular way. The nuclear localization signal can be anywhere in the amino acid sequence of a protein, but they tend to localize towards the outside of the protein where the signal sequence can be seen, and so that other factors can interact with it. And it works in such a way that a protein with a nuclear localization signal can be shuttled into the nucleus, or it can work in such a way that a complex of proteins, right? So basically several proteins that are interacting with each other can be, so an entire complex of proteins can be transported into the nucleus so long as only one of those proteins has a nuclear localization signal, right? Now, the recognition process requires the existence of proteins that are called importans, that are basically nuclear import receptors. And importans, again, have the job of knowing and interacting, binding to the nuclear localization signal on a particular cargo. There are several different classes that recognize slightly different signals, right? So here you have three different cargos, the brown, the yellow, and the green one. And
It's fine.
the each cargo basically has a slightly different looking nuclear localization signal. And there are different importance, which are the blue, the teal, and the pink. I think that's teal, whatever, sage, your choice. And the different importance basically recognize slightly different localization signals. So there are, again, as I was sort of hinting at before, there are proteins that need to go to the nucleus that don't have nuclear localization signals. So what they do is they bind to other proteins. So they bind to partners that do have the localization signal, and that interaction enables them to be shuttled into the nucleus. And that's the case that you see here, right? So you have the important in darker blue. There is the adapter protein in green that has the nuclear import signal. The cargo itself doesn't have a nuclear import signal, but because it's bound to the adapter and the adapter does, then the entire complex can move into the nucleus. Does that make sense? The way that export, so again, we've been talking about importance, so things moving from the cytosol into the nucleus. In the other direction, so from the nucleus out into the cytosol, works very similar. There are proteins that are called exportins that mediate the movement of cargo from the nucleus into the cytosol. All right. Now, there has to be a way to regulate how all of this flux of material happens. And the role of regulating in other words, the main regulators of this trafficking falls on this responsibility falls on the RAN small GTPase family, right? So the RAN small GTPases control the directionality of transport here. We are going to talk a little bit more about small GTPases next week in the signaling lecture, but basically small GTPases work as switch mechanisms, right? So as the name kind of implies, these are proteins, they're small, and they can hydrolyze GTP, right? And they can hydrolyze GTP into GDP. So these proteins basically behave in one way if they're bound to GTP, and they behave in a different way if they're bound to GDP. So the transition between these two states between the GDP form, like this, and the GTP form has to be regulated. And the proteins that regulate that transition are called guanine exchange factors on one side. So RAN guanine exchange factors, or RAN GEFs. This is a protein that, as the name suggests, exchanges GDP for GTP. So in other words, when this protein here is present, it basically kicks the GDP out and it enables GTP to bind, right? That's why it's called an exchange factor, right? And once we have the RAN-GTP protein, the RAN-GTP protein can be recognized by a RAN-GAP protein. And the GAP stands for guanine, or sorry, it stands for GTPase activating protein. GTPase activating protein. So the interaction of the RAN-GAP with RAN-GTP kicks on or turns on the GTPase function of the small GTPase, right? And the GTPase function basically means that the small GTPase, right, RAN, now is able to hydrolyze the GTP that it is bound to, right? So only when this gap protein is there, the GTP, the RAN GTP will hydrolyze that GTP into GDP, right? So that's how we control these the switching between the GTP form and the GDP form. Now, what's important here is that the guanine exchange factors are always nuclear, and the gaps, the GTPase activating protein is cytosolic. So by restricting the localization of the gap and the GEFs, we can ensure that basically we're only going to have RAN-GTP in the nucleus and RAN-GDP in the cytosol, right? And that's sort of what the text on the slide says, right? So GTPases are molecular switches. They switch between GTP or GDP. The transition is regulated by the gaps and the GEFs. RAN gaps are cytosolic, and RAN GEFs are restricted to the nucleus by binding to the chromatin. So we're going to make this a little bit more complicated. So the way that this operates requires us to slow down a little bit. The example that you're seeing here is what happens in nuclear import, right? So the first thing that happens is that you need a protein that needs to be localized to, it's in the cytosol, but it needs to move into the nucleus, right? That's what we call cargo. The cargo is going to be, the cargo has a nuclear localization signal, which is that little darker brown there. And the nuclear localization signal in the cargo is recognized by a nuclear import receptor, by an important, right? So they pair together. The complex of important plus cargo can actually be recognized by the cytosolic fibers that I was telling you. They act a little bit like antennae, right? And once that happens here, there is a process of what we call diffusion, right? In other words, there's a certain probability that the cargo important complex will actually cross the nuclear pore, right? And at some point, it's going to sort of randomly, just by simple chance, it's going to find itself on the nuclear side of the complex, sort of on the basket, right? And once it gets to the basket, it can encounter the GTP form of the small GTPase. So the cargo important complex binds to or interacts with the RAN GTP, and this interaction causes the release of the cargo, because in other words, the important, so the blue molecule here, can either bind to the cargo or to RAN GTP, but not to both at the same time, right? So in the presence of RAN GTP, the important will let go of the cargo and partner up with the RAN GTP, right? And this means that the RAN GTP bound important can now interact with the basket and then basically return back to the cytoplasm or to the cytosol by diffusion. And once it's made its way back to the cytosolic side, here we have the RAN gap, and the RAN gap will trigger the hydrolysis of GTP into GDP. The important likes to be bound to RAN GTP, but not to RAN GDP, right? So it's like RAN GTP has money and RAN GDP is poor, right? So the important wants to hang out with the rich guy, but not with the poor guy. So when this returns to the cytosol and the RAN gap catalyzes or activates the ATPase, the GTPase activity here, RAN GTP will hydrolyze GTP into GDP, and that the important will release the RAN, and it's going to go back to its original conformation when it can bind another cargo and basically do all over again, right? So Alan in the chat is asking, what will happen if RAN GTP is not present in the nucleus? Then you probably won't have you will deplete the cytosol of importance. Because if you don't have RAN GTP on the nucleus, the importance that are out there are going to continue binding to the cargo. The cargo is going to move into the nucleus, but then these importance are going to accumulate in the nucleus and they will not release their cargo. So the importance and the cargo will accumulate in the nucleus, but you won't be able to recycle them. Does that make sense?
Yeah, thank you.
Cool. Thank you. Okay, so this is what happens for nuclear import. What happens with export? It works in a slightly similar way, or in a similar way. It's ever so slightly different. Here you have RAN GTP that binds to the exportin, and the exportin, similar to what happens on the outside, the exportin encounters the cargo. Then the cargo has to have a nuclear export signal, and the exportin bound to the cargo enter RAN GTP, then go through the nuclear pore. Once we get to the outside, RAN gap catalyzes the hydrolysis of RAN GDP, the hydrolysis of GTP into GDP. The system will release the phosphate. And again, remember that these exportin and importance like to hang out with a rich guy, but not with a poor guy. So in the presence of the exportin will not want to be bound to RAN GDP. So at that point, the release of RAN GDP destabilizes the interaction between the important and the cargo. So the cargo gets released as well, right? The free export receptor can now interact with the fibrils, the cytosolic fibrils in the nuclear pore, and return to the nucleus by diffusion, where the free exportin can encounter another molecule of RAN GTP, and the cycle will start over again, right? So the hydrolysis of GTP here causes the offload of cargo, and then the exportin can be recycled back into the nucleus. So there's a question in the chat, Margina or I apologize if I'm butchering your name. How are the RAN GTP and RAN GDP availabilities on both sides maintained? Um, the so the RAN itself, the the RAN the RANs are small proteins that can sort of move um, between the site. Sorry, they can move through the pore because they're maybe 20 kilodaltons, so they are on the smaller side of things. Um, and as I mentioned I think it was in the previous slide, um, the uh, the gaps and the GS are compartmentalized, right? So the uh, the what you are going to see mostly is that in the cytosol you have RAN GDP, and in the nucleus you have RAN GTP. Um, and they simply just they move by diffusion. And if there are things that are, for instance, causing um, an increased rate of traffic from, say, from the cytosol to the nucleus, you're going to get a slight accumulation of RAN GTP in the nucleus. The availability of this RAN GTP is going to depend on the activity of the guanine exchange factors. So there's got to be a certain limit to this that's given by the by the efficiency of these enzymes in catalyzing the release of GDP and the nucleotide exchange. Other than that, remember that the pore itself, I'm actually going backwards. The pore itself is somewhat permeable, and a small thing like the RAN GTPase will sort of diffuse. So this is what, in these figures, these black arrows here basically represent the fact that these things sort of move on their own. They diffuse, and the likelihood of finding them on either side depends basically on what their concentration is. So so that's my best attempt at answering your question. If I don't know if I answered it actually. So did that make any sense to you?
Yeah, that mostly made sense. Thank you.
Thank you. It's complicated. I will acknowledge that. Okay, so the the shuttling between proteins between the nucleus and the cytosol is important is important for a number of functions in specific cell types. So how we enable the exposure of these nuclear localization signals depends on a number of different things. And we are going to exemplify this with this transcription factor called NFAT. So NFAT is basically shuttled between the nucleus and the cytosol. And the cytosolic conformation is inactive. And it's inactive because its nuclear localization signal is actually masked by phosphorylation, right? So there are a number of phosphorylation sites on this transcription factor that when they are phosphorylated, they prevent the exposure of the nuclear localization signal. So the idea is that this particular transcription factor is well known in the activity of T cells, so it's important in immunological responses. So when the T cell becomes activated, so that T cell recognizes a specific antigen and things start happening, one of the things that happens is an increase in intracellular calcium. And this increase in intracellular calcium serves as a signal that activates a phosphatase called calcineurin. And calcineurin actually recognizes the phosphorylated version of NFAT and triggers its dephosphorylation, right? So again, the T cell activates, intracellular calcium goes up, calcineurin is activated, and calcineurin dephosphorylates NFAT. And now um, this dephosphorylated NFAT exposes its nuclear import signal. And then everything that we just mentioned in the previous slide happens, and NFAT now moves into the nucleus, and then it does what it does as a transcription factor. And in the nucleus, there is a lot of ATP, but there are also protein kinases that once calcineurin basically moves away, it enables the phosphorylation of the nuclear localization signal. And when that happens, the export signal becomes exposed. So now we have the export signal, the exportins are going to bind to it, and then we're going to shuttle the transcription factor back into the cytosol, right? So this type of mechanism occurs with many different proteins / transcription factors, or things that are actively shuttled between the cytosol and the nucleus when they need to. Quite often, this is one of the ways in which, for instance, hormone receptors are shuttled between the nucleus and the cytosol, right? And then again, that part of the text is what happens when there is when intracellular calcium decreases. So after the activation of the cell has occurred, right? Any questions? Okay, so now one of the things that characterizes mitosis in eukaryotic cells is that the nucleus actually has to disappear, right? So that we can link the cytoskeleton to the chromosomes, right? So one of the things that needs to happen for the disappearance of the nucleus is actually things have to happen with these lamins, these proteins that basically assemble the nuclear envelope. So lamins are a type of intermediate filament. Did you guys cover the cytoskeleton already? No? So you'll probably hear about intermediate filaments then. So it's a type of fibrous protein. There are many different things. The intermediate filaments in the cytosol are proteins that basically keep the three-dimensional structure of the cell. So they are kind of the beams and poles that keep the cell. They keep they maintain the three-dimensional structure of the cell. Now, the lamins in the nucleus do that for the nucleus, and they interact directly with chromatin in order to stabilize the envelope during interphase, so when the cell is not dividing. In this figure, what you see is basically the organization of these lamins. Again, they form this lattice structure, right? But when the cell is dividing, the nuclear envelope needs to fragment. So this part of the figure here represents what happens in the interphase nucleus. The lamins are these proteins depicted in green that are basically surrounding the nucleus. They are interacting with the nuclear envelope. And there are several signals right before, so at the beginning of the cell cycle, at the beginning of mitosis, that leads to the phosphorylation of lamins. So the phosphorylation of lamins and the nuclear pore complex leads to their disassembly, right? So you phosphorylate them and they start to sort of disgregate. And that means that these proteins are no longer keeping the nucleus in its nice round shape, and the nuclear envelope basically fragments away into chunks of membrane. This happens roughly at the same time as the chromosomes are condensed. And then, so during prophase, the nuclear envelope disappears, the chromosomes are duplicated. But something that's actually interesting is that each chromosome, so during telophase, so later in the process, when the sister chromatids have been separated, sister chromatids basically grab on a piece of, like on a shard of nuclear envelope, and it binds to it. So later on in the process, these lamin fragments are dephosphorylated, and they basically start triggering the assembly of this fragment of the nuclear envelope around each daughter chromosome. And then these fragments fuse around the nucleus and around each chromosome. And then the enveloped chromosomes then fuse together and they reform the nucleus during interphase. This mechanism is RAN-GTP dependent. So this is another thing that RAN-GTP is important for. But we're not really going to go into the details of how that happens. And finally, this is the last thing of this first hour. I'm sorry, I'm going a little late. There are membraneless compartments inside of the nucleus. And the thing that I showed you that we saw before is the nucleolus. This is literally the only difference with the slides that you have is I basically replicated the picture from the first slide. The nucleolus has this function of ribosome biogenesis, right? So new ribosomal RNA is transcribed in the nucleolus, and then the ribosomes themselves are assembled there. But there are other cells in which you can see nuclear speckles, stress bodies, Cajal bodies, or paraspeckles. And as you can see, they have different proteins, different functions. This is just to highlight the fact that there are subcompartments within the nucleus that have specific functions, such as regulation of gene expression, processing of pre-mRNAs, trafficking of small nuclear ribonuclear proteins. That's what NRP means. That are characterized by the concentration of specific molecules, RNA, DNA, and proteins that are associated with a specific function. And in these days, in the research community, the phase separation is all the rage for a number of different reasons. And what happens is basically that there's all of this is the nucleus itself is this sort of aqueous environment, but there are different densities of materials that create sort of subcompartments without the necessity of a membrane. And that's basically what these membraneless compartments are. These are localized concentrations of specific components that are associated with specific functions. Which ones should you know? Absolutely the nucleolus, absolutely the paraspeckles, absolutely the nuclear speckles, the stress bodies. But it's not really a whatchamacallit, it's not a terribly difficult chart, so you should probably just know this. Okay, so that concludes the first hour. Any questions? Yes.
Before, when you were speaking about the GTP process for your exporting, is that the active transport of the energy required, or is that something separate from the ATP required to get big molecules?
Okay, so the question is, when we're talking about this process of cargo movement between the nucleus and the cytosol, this is a type of active transport, but it's not the question was, is this a form of active transport similar to what we find in that requires ATP and such, right?
So like, know that like larger molecules require energy to get
Mhm.
out. I'm asking if this is that energy or if this is just separate to like a recycle that we're in.
Okay, so the question is basically, is this that type of transport that requires energy? And if it requires energy in the same way, right? Am I understanding your question correctly?
I guess is this the only hydrolysis or is there additional?
Gotcha. So we consider this a type of active transport because you need you need a transition between GDP bound and GDP found forms, right? It's not ATP dependent, but you still need GTP and GDP are different currencies of energy in the cell. They're not as abundant as ATP and ADP, for instance, but they work in basically the same way. So for that reason, we consider this a type of active transport. It's just by its very mechanism, it's very, very different from, pumps that directly hydrolyze ATP to move smaller molecules, right? So most transport processes, regardless of where in the cell, that involve macromolecules are considered active transport. The source of energy could be ATP or GTP. And in this case, the source of energy is GTP. Does that make sense?
Yeah.
Sure. Any other questions? Okay, so how about we take 5 and we start in just a second. Okay. So next. No, that's not what I want. Okay. Okay, so the second hour, no, that's not what I want. Sorry about this. There we go. So cell cycle and cell division. There are a couple of parts of this section that are kind of annoying because they're dense, and I'm going to try to skip whatever we can afford to skip. But basically, these are your learning objectives. Keep them in mind as you go over the material. And we're going to start by basically saying a couple of things about the cell cycle itself. The cell cycle is basically the process or the collection of and the sequence of processes that lead to the division of the cell into 2 daughter cells, right? For this to happen, basically 3 processes need to occur. The first is a phase of growth. And during that phase of growth, the genetic material of the parent cell needs to be duplicated. Then the genetic material needs to be segregated into 2 proto daughter cells. And then the cell itself needs to divide, giving rise to 2 daughter cells with a single copy of its genetic material. So we organize this based on a number of hallmarks. This entire process is basically divided in the S phase that involves basically all of the growth, and then the segregation of the chromosome and the division of the cell is it occurs during the M phase of the cell cycle. And the M phase involves mitosis and cytokinesis. Mitosis is the actual segregation of the genetic material. Cytokinesis refers to the segregation of the cytoplasmic components. So this figure shows the different events of the cell cycle divided in phases. Mitosis itself is comprised of 5 stages. And we start with interface first, right? So interface, or an interphasic cell, contains a fully formed nucleus. And this is basically the shape of the cell from the moment that it's born till the very beginning, till basically the beginning of mitosis. The nucleus, the entire nuclear envelope is fully formed. Everything happens, and all the genetic material is duplicated within that nucleus. When mitosis occurs, the first thing that happens is prophase. We're going to talk about the details of each phase a little later on. But maybe when you were in undergrad or even high school, you saw that there were 4 phases in mitosis. Maybe this time around, we're going to add the 5th, which is prometaphase. And there are a couple of important distinctions between prophase and prometaphase. Primarily, the big difference has to do with the state of condensation of the chromosomes, and the stage at which the I don't want to call it really developmental stage, but it's the architecture of the microtubule of the mitotic spindle. But anyway, prophase, prometaphase, followed by metaphase, followed by anaphase, followed by telophase. The transition between metaphase and anaphase is crucially important for the development of the cell cycle. We're going to talk later about why. And cytokinesis actually begins before mitosis ends. So there's a certain degree of overlap between the end of anaphase and the beginning of telophase. And at this time, the cell starts to segregate its cytoplasmic materials. And in the end, what you get is 2 daughter cells. And these daughter cells basically go into interphase. There's the phase of growth. Then there's the phase of DNA replication. And then we go back to the origin. This transition between metaphase and anaphase is really important. Cells need to make sure that everything is in its place before moving into anaphase. So the cell, if the cell basically does sort of a check, it makes an inventory of everything that it has, and it makes sure that it has enough for the daughter cells, for each daughter cell, and it makes sure that there's enough DNA in each daughter cell. If there's not enough, so if there's any sort of imbalance there, the cell can and will pause there, and it will arrest its cell cycle until everything gets sorted out. Once it gets sorted out, the cell will commit to finishing mitosis and go through with cytokinesis. All right. So we're going to say a few words after later on about the transition between metaphase and anaphase. So the distribution of genetic material depends on the microtubules that form a structure that's called the mitotic spindle. So in prophase, the first stage of mitosis, what we have is a relatively intact nuclear envelope, right? The chromosomes have started condensing. So the chromatin starts condensing further and further. And what we get are like proto-chromosomes that are not fully condensed yet. But what characterizes this is the presence of an early mitotic spindle that begins to form. It forms starting from these centrosomes, which are microtubule organizing centers, and they trigger and direct the polymerization of these microtubules that are forming the spindle. On this figure to the right, what you get is the microtubules are stained in red, and the DNA is stained. The microtubules are stained in green. The DNA is stained in red. And that's actually a real cell. Prometaphase is characterized by the fact that the mitotic spindles have arrived to the poles of the cell, right? So the centrosome in prophase, the centrosomes were sort of migrating towards the poles of the cell. By prometaphase, they have already arrived there, and the nuclear envelope has fragmented, and the chromosomes are finally fully condensed. And on the right, you can see the difference, how different they look, how different the cells look between prometaphase and prophase. Something that's important to mention is that the mitotic spindle contains different types of microtubules. Some microtubule fibers are called kinetochore microtubules, and those are the ones that are seeking to bind to the actual chromosomes. So we're going to say a couple of words about them later. There are the astral microtubules as well that are basically facing away from the chromosomes. During this process, basically, once the chromosomes attach to the kinetochore microtubules, the chromosomes are actually moving. They are sort of trying to find their place. And the place that they're actually trying to find is the metaphasic plate, right? Which is something that some people call the equator of the cell. At metaphase, basically what you get is that the chromosomes that are fully condensed are attached to kinetochore microtubules on both ends. So a metaphasic chromosome, a metaphasic duplicated chromosome, has one kinetochore microtubule attached on one side and one kinetochore microtubule attached to the other side. So they are effectively connected to both poles of the cell, right? They're physically attached to both poles of the cell. Each one of those metaphasic chromosomes is attached to one kinetochore microtubule from each pole, from each centrosome. At metaphase, the spindle microtubules are arranged in a relatively particular fashion. So you have the kinetochore microtubules in blue. And again, this chromosome has kinetochore microtubules to each pole. Then there are the astral microtubules that are basically facing sort of away. And then there are the interpolar microtubules that basically bind to each other, and they interact via motor proteins. So the motor proteins are those little black balls that you see in between, right? So I want you to keep this organization in mind, because it's important to understand what happens later. These motor proteins are controlling what happens to the microtubules in the spindle. There are you are aware that there are two general, well, maybe not, but you will hear about this later. There are two kinds of motor proteins. There are dyneins and there are kinesins. They are characterized by the fact that they move towards different ends on the microtubules, right? So in this case, what you don't need to remember from this, you don't need to remember the different specific kinesins. So I'm not going to ask you about kinesin 14 versus 5 versus 4 versus 10. But what I do want you to see is that these motor proteins are localized to different places in the spindle, and because of their action, they can actually physically move they can generate displacement within the spindle. And that means that displacement can generate forces, right? And those forces ultimately are the ones that pull on the chromosomes, right? So anaphase is the phase that requires or that causes the separation, the actual segregation of the sister chromatids. So the first requirement for anaphase is a stable spindle, right? So a spindle that is attached to each chromosome on both kinetochores by kinetochore microtubules. And it also requires the degradation of a set of proteins called cohesins. So the first thing that and this is a really, really, really important concept, the attachment of the microtubules to the kinetochores needs to be stable. So a stable arrangement is one in which both kinetochores in each one of the sister chromatids is attached to a kinetochore microtubule, right? And the idea is that the process of metaphase, and one of the reasons why metaphase is the longest phase or the longest stage in during mitosis, is that initially most chromosomes look unstable like this. There is one kinetochore is attached to to a kinetochore microtubule, but the other one is not, right? So during metaphase, what we want is for that unstable conformation to become stable. And it only becomes stable, whoops, I'm actually going back. It only becomes stable once you get this conformation in which each kinetochore in each separate sister chromatids is attached to a microtubule from two different poles so that force can be applied equally on both sister chromatids. Attachment of both kinetochores to microtubules from the same centrosome is unstable. Attachment of the same kinetochore by microtubules from two separate centrosomes is also unstable. The only thing that the cell wants here is this: one kinetochore attached to one pole, to one centrosome, the other to the opposite pole of the cell. This is super, super important because if you don't have this arrangement, so if you find yourself in any of these other unstable conformations, you're going to get an uneven separation of the sister chromatids. And if this happens in a germline cell, right? So in a cell that is going to generate, say, an egg or a sperm cell, what you're going to get is something that could be, for instance, a trisomy, right? You could get 3 copies of a particular chromosome in a cell, right? And the majority of those situations are actually non-viable, right? There are a few trisomies that are viable. Trisomy 1821 are the ones that come to mind, but there are others. Now, stable attachment to the kinetochore is the first requirement. The second is the activation of a protein complex called the anaphase promoting complex, or APC. The APC is found in an sort of unstable conformation. Sorry, inactive conformation, not unstable. And at some point because of signals that change their levels within the cell at different stages, there's this cyclin called CDC20 that is able to activate the APC. The concentration of CDC20 increases at the beginning of anaphase, right? And there are a number of reasons for that. The activated APC then causes the degradation of another protein called securin. Securin is an inhibitor of an enzyme called separase. So when there is no anaphase, so before anaphase starts, separase is kept in an inactive state by binding to securin. But when anaphase starts, activation of the APC, causes the degradation of securin, the activation of separase, and separase, all right, separase then is able to degrade a protein complex called cohesin, which holds both sister chromatids together, right? In other words, if we think back to the S phase of the cell cycle, when the nuclear material is being duplicated, then that duplicated material has to basically come together into the condensated chromosomes. And at the beginning of mitosis, the chromosome is duplicated, so there are two sister chromatids. Those two sister chromatids are always sort of glued together. That glue that holds those sister chromatids together is called cohesin. So that in order for those sister chromatids to be segregated to their future daughter cells, we have to basically remove the glue. And that's the job of separase. So separase basically gets rid of the glue that's keeping those chromatids together. When the glue that holds those chromatids together is gone, now the microtubules that are bound to those kinetochores are exerting forces, and those forces can now pull the sister chromatids away from each other, right? That's what happens here, right? So that's the job of this anaphase promoting complex. Does that make sense? Any questions so far? Okay. So the actual segregation of chromosomes occurs via two separate processes that happen kind of simultaneously. One is called anaphase A. And in anaphase A, basically what you have is that the kinetochore microtubules, so the microtubules that are attached to the sister chromatids, those specific microtubules shorten. So you can see that in this form, the centrosomes are basically on the same place, right? They're kind of, they stay static, and what moves are the actual sister chromatids. So the forces are generated here at the kinetochores. The other form in which anaphase occurs is called anaphase B. And this happens because the interpolar microtubules that have all of these motor proteins in them, those interpolar microtubules are basically starting to slide away from each other. In these cases, the kinetochores, the kinetochore microtubules don't really shorten that much, right? But what's going on is that the poles of the cells, of the future daughter cells, are basically pulling away from each other, right? So in this case, the activity of the motor proteins at the interpolar microtubules pushes both centrosomes away, and as these centrosomes move away from each other, they drag the sister chromatids with them, right? So again, you can either generate the force directly on the kinetochores, and that's what characterizes anaphase A, or you can generate the force at the interpolar microtubules and basically move the centrosomes away. All right? Those are the two forms in which anaphase occurs. Now, once we have ensured that the genetic material is distributed, then we enter telophase. During telophase, we have the nuclear envelope begins to reform. The interpolar microtubules continue to sort of interact, but then we have the beginning of formation of a contractile ring, sort of in the place where the cell is going to divide, where the cytoplasm is going to divide. And during cytokinesis, we have the complete reformation of the nuclear envelope. So during later stages and later times in cytokinesis, the nuclear envelope is completely reformed. You get one centrosome per daughter cell, and the contractile ring is basically squeezing the cytoplasm at this particular location. This contractile ring is made of actin and myosin. So these are the contractile proteins that you find in other cell types. There are specific types of myosin in particular that form this contractile ring. And finally, you get this leads to the pinching off of the two different cells, and you get two daughter cells, right? Something that's also relevant is that so far we talked about mitosis. Meiosis is a specialized form of cell division that only occurs in the gonads. So these are the tissues where reproductive cells are formed, right? Reproductive cells are particular because we have to ensure that they get only half of only half of the genetic material of the other cells in the body, right? So these reproductive cells are called gametes. And something that's actually important to remember is that the cells that are going to become gametes have to go through regular mitosis before they start meiosis. So they go through a full round of DNA replication. Once the so let me put it this way: the first stage involves the full mitosis of this initial cell, right? This is a progenitor cell. This cell is going to undergo mitosis, and you're going to get two daughter cells. One of those cells is going to enter meiosis 1. And during meiosis 1, what you get is that first you're going to get recombination of the genetic material. So there's going to be a pairing of the chromosomes, and there's going to be exchange of segments, and all sorts of fancy stuff is going to happen in these cells. And then during metaphase, all of that pairing happens during the metaphase of the first meiotic division. But then anaphase is going to cause the segregation of homologous pairs, right? What does that mean? In regular mitosis, what you separate are sister chromatids, right? Copies of the exact same genetic information. In meiosis, in the first metaphase in the metaphase stage of the first meiotic division, what you're going to do is you're going to pair the homologous chromosomes. One comes from one parent, the other one comes from the other parent. Those homologous chromosomes have exchanged genetic material. So at that point, once recombination occurs, they are not identical, right? And you have them paired, right? During the first meiotic division, these are duplicated homolog chromosomes. Duplicated homologous chromosomes. So each of these homologous chromosomes contains two sister chromatids, right? During the first anaphase, so anaphase 1, these duplicated homologs are going to be segregated, right? Because we are segregating duplicated homologous chromosomes, we are actually halving. We're splitting the genetic information in half. And at the end of the first meiotic division, you're going to get haploid cells. These haploid cells contain duplicated chromosomes. So basically you have 20, in the case of humans, each daughter cell is going to have 23 chromosomes, not 46. Those 23 chromosomes are duplicated, so each one of those chromosomes contains two sister chromatids. So the daughter cells of the first meiotic division are said to be haploid. During the second meiotic division, what you're going to do is you're going to segregate the the sister chromatids from those individual homologous pairs. So starting from a single progenitor, what you usually get is 4 different daughter cells at the end of the second meiotic division. Each of these cells is genetically distinct from the other, right? And this is enabled by the fact that there's no S phase in between the first and second meiotic division. So at the end, you exit the first meiotic division and go straight into the second. So the second meiotic division is actually almost identical to a regular mitosis. You start with duplicated chromosomes. During metaphase, you align those duplicated chromosomes in the equator of the cell. During anaphase, you separate the sister chromatids, right? And that's the big difference between the second and the first meiotic division. In the first meiotic division, you separate homologous pairs, not sister chromatids, all right? Does that make sense? Any questions? All right. So the cell cycle comprises 4 main phases. M phase is the mitosis phase that we just described, but then the interphase contains G1, S, and G2, right? G1 and G2, so the G comes from from gap, because this is the name that people decided to give these stages. So they're called gap 1, gap 2, and in between gap 1 and gap 2, you have S, which is synthesis, S for synthesis of DNA. Now, this graph is basically a representation of the relative amount of DNA per cell, right? And the number of cells that have that amount of DNA at any particular time. So again, on the y-axis, you have number of cells. So this is just a count of cells. And on the x-axis, you have a relative amount of DNA, meaning 1 full copy of the genome or 2 copies of the genome. So basically, this large peak represents cells that have not duplicated their DNA. This smaller peak represents cells that have duplicated their DNA. So you can confidently say that all of these cells that are in this large peak here are in G1, because G1 is just growth. The second peak happens after you've duplicated your DNA. So those cells are in G2. And everybody in between are somewhere along the S phase. They are duplicating their DNA, so they have somewhere between 1 and 2 copies of their genetic material, right? At different stages, it depends, it takes time for cells to duplicate their DNA. So if you have a population of cells that are not synchronized, you're going to get a certain number of cells that are going to be in G1, a certain number of cells that are going to be duplicating their DNA, and they're going to be at different places along that process. And then you're going to get a bunch of cells that have already duplicated their DNA, right? That's basically what this figure is telling you. Martina is asking whether there is a G0 phase. G0 is sort of a phase of quiescence, right? G0 is a state in which cells are not dividing anymore. So those are cells that are basically said to be outside of the cell cycle. And some cells can go into G0, live there, and then go back into the cell, go back to the cell cycle and continue dividing under certain circumstances. But for the sake of this lecture, we're going to ignore them. Okay, so the progression through the different stages of the cell cycle is very tightly controlled. And the control here basically comes in the shape of checkpoints, right? So there are 3 main checkpoints in the cell cycle. There is the start transition that happens at the end of G1. The start transition basically is the cell asking itself, do I have everything that I need in order to proceed with DNA duplication? Is it worth it? Do I have nutrients? Do I have all the signals that are necessary for me to start this process? The second checkpoint is at the right at the end of G2 and the beginning of mitosis, right? The cell is asking, is all the DNA replicated? Is the environment favorable, right? Again, this process takes energy. Do I have nutrients? Do I have everything that I need? But most importantly, do I have exact two copies of my genetic material? If you don't have two exact copies of your genetic material, then if you go through the cell cycle, you're going to get two daughter cells that are not going to be identical, right? So if the cell has not finished replicating everything, or if something has been damaged that hasn't been repaired yet, the cell is going to stop there. And finally, the third checkpoint happens at the metaphase to anaphase transition. And here the key question is, are all the chromosomes attached to the spindle, right? If I proceed, will my two daughter cells get the exact same information, right? So these cyclin, the regulators of each one of these checkpoints are basically complexes that are complexes of proteins called cyclins and cyclin-dependent kinases. So cyclins and CDKs, right? There are several of these. So generally speaking, there are more, but we're only going to really care about these four. So the G1 CDK, the G1S CDK complex, the S CDK, and the M CDK. These complexes increase their expression and their activity at different points along the cell cycle. And they are basically formed of particular combinations of cyclins and CDKs, right? So each complex has a different cyclin. The CDK partners can be shared, right? So you see the CDK2 and CDK1 are present in two different complexes, right? So do you need to remember the details of all of this? No, I wouldn't say, I don't think so. However, I would suggest that you pay attention to the cyclins, right? So the cyclins are important here. Meaning understanding which cyclin goes with which complex is relatively important. So this graph tells you the relative levels of expression of each complex at different stages in the cell cycle. So you see that these dashed lines represent the main checkpoints. Those checkpoints are places where these cyclins become really, really important. So at the start checkpoint, you get, the G1S cyclin has been active for a while. But at this point, the G1S, the activity of the G1S CDK complex triggers the activation of triggers the increased expression and activity of the S CDK complex, right? So the S cyclin increases. When the S cyclin levels are high, that causes the turning off of the G1S CDK complex. And the S cyclin, the S CDK complex basically sustains the occurrence of the S and the G2 phases. By the time that we get to, after DNA has been duplicated, there's a gradual increase in the expression of the M cyclin, right? And when the M cyclin reaches its peak activity, that is when the G2M transition takes place. The mitosis is basically initiated, and that triggers the degradation of the S cyclin. S cyclin activity goes down, and then the M cyclin sustains the first three phases of mitosis. So you have prometaphase, metaphase, and then you get to the metaphase anaphase transition, right? And at this point, the high activity of this M-CDK complex is what triggers the activation of the anaphase transition, the anaphase promoting complex and whatnot. We're going to say a couple of words about that in a bit. This is what I was referring to earlier as this big transition, this big biochemical transition in the cell, right? During or at the metaphase anaphase transition. So what do these things do? The increased activity of the SCDK triggers activates the DNA replication machinery. That kind of makes sense. At the beginning of the S phase, what you need is to replicate the DNA. The MCDK becomes activated, and its main job is to trigger the expression and activity of the mitosis machinery, right? The proteins and factors that are important for mitosis. The regulation of cyclin CDK, is mediated by phosphorylation and interaction with other proteins. So basically, here what you have is an inactive kinase, an inactive CDK, right? The interaction of the kinase, of the CDK with its cyclin triggers a partial activation of the complex. And then there is yet another step in which another protein involves here. It's called, and it's a kinase that phosphorylates here, triggers the phosphorylation of a segment in the kinase, and that triggers the full activation. The take-home message here is that the activation of this requires the binding of the cyclin and the phosphorylation of the kinase for the whole complex to be fully active. These cyclin CDK complexes can be inhibited by interaction with other kinases, right? So, for instance, the active cyclin CDK complex can be phosphorylated in a particular residue here by WEE1, and WEE1 basically turns the kinase off, and that turns the whole complex off. That inhibitory phosphate can be removed by a CDC25 phosphatase, right? So forget about the details of this. The important take-home message here is that we can phosphorylate and dephosphorylate these proteins to turn them on or off, right? And there are cell cycle regulator proteins that can basically bind to the full complex and trigger their inhibition, right? So in this case, P27 is an important inhibitory protein here that binds to the full complex, and it triggers its inhibition, right? These are examples. Again, what I want you to see here is not the details, not the specific names of any of these proteins or factors, whatever. What I want you to basically focus is on the general idea of the process, right? Again, activation by phosphorylation, inhibition by phosphorylation, and dephosphorylation, inhibition by binding to proteins, right? So the examples, what activates and what inhibits, those are specifics, but I'm not necessarily going to ask you to remember those details, okay? Any questions so far? Okay. So the way in which the cell cycle is regulated basically depends on the synthesis and degradation of the proteins that form these cyclin CDK complexes, right? So this is complicated. Not really going to go in too much detail of it. But for instance, in this case, if you remove if you remove an inhibitor, right? so this cyclin kinase inhibitor, so it's a CDK inhibitor protein. If you basically remove this inhibitor, then you allow the expression of the G1S complex, right? And that commits the cell for cell division, right? That degradation requires something that's called the proteasome. Maybe you have heard about proteasomes before. If you haven't, the proteasome is basically like a shredder. If you at home have a shredder for like a document shredder, the proteasome is a protein shredder. And you can target proteins that you don't want, but you can send them basically to the proteasome, to the shredder, by tagging them with ubiquitin, which is sort of a protein tag. The ubiquitination, so the addition of this sort of death tag on these proteins, depends on proteins that are called ubiquitin ligases, and SCF is one such example that does that, right? So, or that basically triggers the activation of these ubiquitination enzymes. Again, forget about the details. What's important is that the degradation of the inhibitor leads to an increase in the expression of the cyclin CDK complex, and that triggers the commitment to cell division in this case. As we were saying before, one of the things that the anaphase promoting complex does is it makes sure that there's this transition between metaphase and anaphase, right? But the activity of the M cyclin, that, remember, the M cyclin becomes important here to trigger the mitosis machinery. The M cyclin, the M CDK complex here, can also be degraded, and the individual that leads to this degradation is the anaphase promoting complex, right? So when the anaphase promoting complex becomes active, it triggers the tagging of the M cyclin, and that leads to its degradation. Does that make sense? Any questions? Okay. I'm going to sort of skip this because it's a little complicated, and it's a relatively low yield complex concept. But basically, when DNA replication along the cell cycle, when you're starting DNA replication, what you need is the activation of the S cyclin complex, and basically, what happens is that the replication machinery needs to be sort of pre-assembled at the replication origin sites on the DNA. And what the S cyclin complex does is it triggers the activation of these replication complexes. So in other words, an increase in the activity of the S-CDK complex triggers the activation of the DNA helicase, and that ultimately triggers replication, right? So again, you assemble all of the machinery where it needs to go, and CDK, the S-CDK complex basically gives the go for replication to occur, to start. And this next slide basically tells you a little bit more in detail about what each step involves. I'm going to, I'm not really going to mention too much. If you want to, if you are curious, you can read about this. I very much doubt that I'm going to ask you about this particular process, okay? In the interest of time, we're gonna just move forward. Now, the activation of the M CDK complex is what triggers the entry into mitosis. Remember earlier, I told you that the M CDK complex is the one that basically triggers the activation of the replication machinery. Sorry, the mitosis machinery. So here, the cyclin-dependent kinase that's involved in this process is CDK1, right? So when CDK1 binds to the M cyclin, and this forms an inactive component, right? And an inactive complex. That's what I was thinking. Now, the idea here is that there is an activating kinase here, right, called CAK. And this CAK kinase basically triggers the phosphorylation of this complex at this activating phosphate right here, right? Now, simultaneously, Wee kinase, which we talked about before, Wee kinase also phosphorylates the CDK molecule at the inhibitory site. So at this point, this complex remains inactive because it's phosphorylated twice, in the activating and in the inhibitory phosphate. Then we get the activation of this phosphate called CDC25, and the CDC25 basically removes the inhibitory phosphate and triggers the full activation of the complex. This active MCDK complex triggers a positive feedback by which it causes more activation of the phosphatase, right? Leading to basically more activation of the MCDK complex. And then the active MCDK also inhibits the inhibitory kinase, further helping this process to go forward, right? So again, the initial activation of the initial formation of the MCDK complex is sort of self-sustaining, because as you go through the process, you first phosphorylate twice, and then you dephosphorylate at the inhibitory site. That triggers activation, and the activation of the complex basically activates further, or it basically helps. What's the word that I'm looking for? It further activates the factors that are causing this activation, and that's what we basically mean by positive feedback. More CDC25, that means more dephosphorylation of the inactive of the inhibitory phosphate, and it means more inhibition of the Wee kinase. So that means basically you get more activation at the right places. Any questions? I would like to. Let me see one thing. Okay, so now what causes a cell to enter into the cell cycle, right? So the entry into the cell cycle has to be sort of regulated, right? And the factors that tend to cause this are called mitogens, right? Mitogens, the word basically means they stimulate mitosis. These are signals, usually extracellular signals in the form of growth factors or hormones or what have you, that lead to increases in the activation of the cyclin CDK complexes. So for instance, the one classical activator of the cell cycle is the RAS-MAP kinase pathway. So usually a mitogen, say, growth factor, binds to its receptor, and the receptor triggers the activation of an intracellular GTPase called RAS. And activation of RAS causes a sequence of events that lead to the activation of a kinase inside of the cell that is called MAP kinase. And MAP kinase then activates further things downstream. All of these are like, sequences of cascades of biochemical events Ultimately, this kinase goes into the, is actually transported into the nucleus, and it triggers phosphorylation of several transcription factors that will lead to gene expression. The genes that are activated by this pathway are called immediate early genes. One of these immediate early genes is called MYC, and MYC is a master transcription regulator. Activation of MYC triggers the expression of delayed response genes. And these delayed response genes involves the expression, increases in the expression and therefore the activity of the G1 CDK complex. And when G1 CDK complexes are activated, this triggers a bunch of different things that lead to basically the activation of the S-CDK complex and then the DNA synthesis, right? So the details here are not really super relevant. Again, one of the things that is important here is the activation of E2F. Activation of E2F causes increases in protein translation, and then it leads to transcription of the S-phase genes. Once the S-phase genes have expressed, you get basically the cell is cohabitated by the G1S complexes and the S complexes. The S complexes then take over, and then you get DNA synthesis. So all of this is to say, these cyclin CDK complexes that we were talking about, they don't happen in the void. They don't happen in a vacuum. They respond to signals that come from the outside of the cell that are basically saying, this is your time to start dividing, right? Now, there are also situations that can lead to the arresting of the cell cycle, right? So a typical example is DNA damage. So when DNA damage occurs, the cell needs to make sure that it stops anything that it's doing so that it can focus its resources into repairing that damage. For instance, cells that are exposed to X-rays, right? The X-rays can cause physical damage to the DNA. So breakage in the double helix, substitution or elimination of nucleotides, is a bunch of different really weird stuff that can happen when you expose cells to X-rays. And there are proteins that recognize this. So the DNA, the nucleus has a bunch of surveillance proteins that are constantly monitoring whether things are okay. When there is something that shouldn't be there, like damage, these factors basically become activated, and they lead to the phosphorylation of a protein called P53. P53 normally is kept in an inactive state by this protein called MDM2, and MDM2 basically sends, it tags P53 with this death signal that we call ubiquitin. But when this damage happens, P53 becomes phosphorylated. The phosphorylation of P53 leads to the dissociation from MDM2, and it stabilizes the active conformation of P53. And this phosphorylated P53 is able to trigger the transcription of a gene called P21. And P21 is this inhibitor protein, right? The CKI proteins are proteins that bind to the cyclin complexes and prevent them from doing their work. So activation of P53 triggers expression of P21. P21 is a CKI, and then as P21 becomes active, it triggers the inactivation of the G1S complexes, and that stops the cell cycle. And it basically buys time for the cell to repair the damage that has occurred before the cell can actually move into replicating the DNA, right? It's super important to understand that this causes the inactivation of the G1S complex, because the G1S complex is the one that's driving the GS phase, and it basically commits the cell to the S phase, right? We need to stop the S phase from occurring so that the damage to the DNA is repaired. Once the damage of the DNA is repaired, then you get dephosphorylation of P53, degradation of P21, and then the cell can continue into the next phase. Does that make sense? Okay. Any questions? And this is just a summary. These, the different complexes that are important. You should be able to look at this slide and say, okay, this is what's happening at each step, right? So when the extracellular environment is favorable, you get expression of G1-CDK. So this initially causes increases in the G1 cyclin and the S cyclin. The S cyclin then takes over. It triggers the entry into the S phase. Then G2 happens. During G2, the MCDK complex becomes active. That triggers the entry into mitosis. And then it also triggers the initiation of the expression of the anaphase promoting complex. The anaphase promoting complex basically shuts everyone down, and that leads to the conclusion of mitosis, right? So that's ultimately what you want to look at, and these are inhibitory factors to each of them. Okay, so let's take 5, and at 8:30 we're gonna say a few things about apoptosis and cell death. All right, so the last part of today is focused on apoptosis. Maybe you have heard about that concept before. It refers to forms of cell death. It's not the only form of cell death that we're gonna talk about. So these are your learning objectives. Make sure that you understand them. And we're gonna start by comparing the two main forms of cell death, apoptosis and necrosis, right? This is an important chart for you to know. Apoptosis works on individual cells. It's characterized by the shrinkage of the cell and its fragmentation. It's characterized by the what we call the blebbing of the membrane, and the plasma membrane of a cell basically remains continuous, right? So it's a relatively orderly form of cell division. From a biochemical perspective, it is characterized by the flipping of phosphatidylserine. Phosphatidylserine is one of the many phospholipids that forms the lipid bilayer, but in healthy, normal cells, it is usually localized to the inside of the membrane, right? So in the lipid bilayer, it's facing the intracellular environment, right? So when you get flipping of phosphatidylserine, so when you can find it facing outside, that is a sign of apoptosis. The activation of caspases, which are the proteins that mediate apoptosis leads to basically the breakage of mitochondria, which then release their contents. So primarily calcium, and an important marker of apoptosis is the release of cytochrome C, which is a mitochondrial protein. Organelles contract, the chromatin becomes fragmented, and the cleavage of the DNA happens in between the nucleosomes, so in that linker DNA segment. And ultimately, the cell is phagocytosed away by resident macrophages, and all of this requires ATP. So this is a control process. Necrosis is sort of the direct opposite of all of this. It involves groups of cells. So necrosis doesn't happen cell by cell. It happens to many cells simultaneously. And the key thing is that the loss of cellular integrity leads to the swelling of the cell. So the cell ultimately bursts, right? It lyses. And the mitochondria swell. The organelles also swell as opposed to contraction. The nuclear envelope is disrupted. And the cleavage, the rupture of the DNA is random, not at those nice linker fragments. The cells usually so necrosis is usually something that is dampened by an inflammatory response. So it causes an inflammatory response, whereas apoptosis does not trigger inflammation, or not necessarily. And then the process is passive. So necrosis is basically the cell has stopped working and it just like explodes, right? So again, important to understand this chart. Apoptosis depends on the activity of a family of proteins that are called caspases, right? And there are several types of caspases. The two main types of caspases are called initiators and executioners, right? So initiator caspases, as the name sort of implies, are the ones that trigger the process, and they become activated in response to apoptotic signals, whatever the nature of those apoptotic signals is. These initiator caspases, and here the most common examples are caspases 8 and 9. Each one of these proteins is found as a monomer, so a single protein chain. They have an adaptor binding domain and a protease domain. And within the protease domain, there are cleavage sites, right? So the apoptotic signal causes the activation of these adapter proteins, that cause the dimerization of the caspases, right? So they form this dimer. The dimerization activates their protease domains, and the protease domains basically means that they sort of self-cut, and that means that the cleavage finalizes their activation, and what you get is basically this large subunit bound to a small subunit that is active. These initiator caspases are sometimes called pro-caspases, right? Particularly in their inactive state. And then again, these apoptotic signals trigger the assembly of the assembly and activation of the full initiator caspase. Now, this rearrangement triggers the rearrangement of the protease domain, basically, triggers this full activation, and the activation, the active initiator caspase triggers the cleavage of executioner caspases, which normally are found in this inactive state. Here, these are caspases 3, 6, and 7. So the active initiator caspases cuts the executioner caspases at their cleavage sites, and it leads to the formation of an active executioner caspase. And these executioner caspases, when they're fully active, they cleave multiple things at sort of simultaneously. And the breakage of these targets is what ultimately triggers apoptosis, right? So again, we have initiator caspases that are inactive, and then when something tells the cell, hey, you need to die, this triggers the activation of the initiators. The initiators activate the executioners, and the executioners are the ones that do the dirty job. This happens in an amplification cascade of sorts. So you have one molecule of an initiator caspase, triggers the activation of many executioner caspases, and the many executioner caspases then triggers the activation of even more caspases downstream, and then you basically get, chopping galore all over the cell, right? And examples are that, example is that the first set of caspases will trigger the cleavage of cytosolic proteins. Then the nuclear lamin is sort of protected because the lamin is intranuclear, so it's sort of protected by the nuclear envelope. So in order for that protein to be cleaved, you need to activate many, many more things that basically are going to start macheting left and right through the cytoplasm till they get to the nuclear envelope and they can make it into the nucleus before they can they can cleave the nuclear proteins, right? So there are two apoptotic pathways, right? So these are pathways that mediate the activation of these these initiator caspases. The extrinsic pathway is the first one that we're going to discuss. And it's mediated by cytokines and cytokine receptors, right? The tumor necrosis factor family of ligands, and maybe you have heard about TNFs. The TNFs are very potent pro-inflammatory agents, but they can also be pro-apoptotic factors. So the idea here is that there is a particular protein called Fas ligand. And this Fas ligand is a transmembrane protein found on the membrane of immune cells, primarily T cells, and also killer lymphocytes. And this Fas ligand protein helps this killer lymphocyte to basically, it enables the lymphocyte to dock to its target cell using the Fas death receptor, right? Now there are other examples, like TNF binds to the TNF receptor, and there are a bunch of other things that happen here. But the Fas ligand is the one that we're going to focus on for now, right? So when Fas ligand binds to its receptor on the membrane of a target cell, right? And this target cell could be, for instance, let's say, it could be a cancer cell. It could be a cell that has experienced DNA damage, and that damage is beyond repair. There are many possibilities. But when the ligand binds to its receptor, it leads to the receptor trimerization. So you have the individual receptors. In the presence of the ligand, the receptors are going to come up in triads, so 3 receptors together. And the activated Fas receptor is going to recruit its intracellular domain. And the intracellular domain is going to trigger the recruitment of initiator caspases, right? And this leads to the assembly of DISC, right? The death-inducing signaling complex. And basically this means Fas ligand binds to the receptor triggers the recruitment of all of these initiator caspases in the form of DISC. And then DISC is going to trigger the activation of the caspase of the protease domain here, and it's going to lead to the activation of caspase H, which, remember, is an initiator caspase. Once the activated initiator caspase becomes active, that leads to the activation of the executioner caspases, and then all hell breaks loose in a controlled fashion, though, inside of the cell, and the cell is going to start dying. And when this happens and the lymphocyte is happy, the lymphocyte just walks away, right? So basically, this is like the lymphocyte giving the kiss of death to its target, right? It's called the extrinsic pathway because this requires a stimulus that is outside of the cell, right? The intrinsic pathway is initiated by mitochondria. inside of the cell, right? So it can be triggered by several different factors. Mostly these represent metabolic imbalances, so or biochemical imbalances in the cell. So, if there's too much calcium, calcium is great, but too much of it is really bad. Excess oxidation, so if there's excess oxygen radicals, or the presence of pro-apoptotic proteins such as Bax and BID, right? So the key thing that happens is that when there are apoptotic stimuli, the mitochondria becomes unstable and it leads to the release of cytochrome C. I told you before that release of cytochrome C is sort of a hallmark of apoptosis. Released cytochrome C is basically the signal for the activation of this complex called APAF1. And APAF1 binds to the cytochrome C, and that leads to the assembly of this really large seven-member complex called the apoptosome. And the apoptosome is able to recruit it's able to recruit caspase-9, right? And caspase-9 is another one of these initiator caspases. So the apoptosome recruits and triggers the activation of caspase-9, and then active caspase-9 will lead to the activation of executioner caspases, and then the full-blown apoptosis. Then again, the thing that you need to remember from this is this depends on the release of cytochrome C from the mitochondria. We're going to mention this a little later, but there are anti-apoptotic factors in the cell that basically usually prevent this from happening in the absence of anything going wrong. So when everything is okay, there's this protein called BCL2 that blocks. It interacts with cytochrome C and prevents it from being released. So speaking of BCL2, BCL2 is a family of factors. BCL2 is one such member of this family. And some of them are pro-apoptotic. The others are anti-apoptotic. And they regulate the intrinsic pathway, right? They don't really do anything to the extrinsic pathway, but they do modulate the intrinsic one. So in the absence of apoptotic stimuli, so when things are okay and nothing is going wrong, basically BCL2, BCL2 and other anti-apoptotic members, such as BCLX, they are basically inhibiting the effector members of the family. So things that are important, not necessarily super important, but all of these proteins are characterized by the presence of this BH domains in their sequence. So BH 1, 2, 3, and 4. BH means BCL homology domain. And ultimately what they do is that the anti-apoptotic are active and they inhibit the effector proteins. So this basically allows the cytochrome C and all mitochondrial contents to stay inside of the mitochondria. When we have pro-apoptotic stimuli, so proteins like Bax and BAC, what this does is they overcome the inhibition by BCL2, and they lead to the activation of these effectors that basically create large pores on the outer membrane of the mitochondria. And via these large pores, they allow the release of mitochondrial intermembrane space components, right? So again, in the mitochondria, we have these effector proteins. These effector proteins are kept at bay by the BCL2 anti-apoptotic family members. But in the presence of pro-apoptotic, they will actually assemble together and they create this pathway for the release of mitochondrial content. And there are some examples of pro-apoptotic proteins that only have BH3 domains that basically do this in a sort of a targeted way. So for instance, BAD, BIM, BIT, PUMA, and NOXA, again, don't necessarily remember the details of those, but what they do is they trigger the activation of this BH3 only protein, and this protein basically inactivates the anti-apoptotic and activates the pro-apoptotic members of the family. So long story short, this is a family of important regulators of apoptosis. Some of them prevent it, some of them promote it. And their relative activity is what regulates all of this. The intrinsic and extrinsic pathways of apoptosis are not independent of each other, right? So there is sort of a certain degree of integration between the two. So activation of the Fas ligand of the Fas receptor by Fas ligand triggers the activations of the initiator caspases, the activation of the executioner caspases, and then they trigger all sorts of things, right? But these executioner procaspases can actually interact with members of the BCL family, particularly the pro-apoptotic members. So executioner caspases activated by the extrinsic pathway can lead to the activation of the activation of members of the intrinsic pathway that are going to cause a release of cytochrome C. If cytochrome C is released, then the apoptosome becomes active, and the apoptosome, remember, is basically a big initiator caspase that ultimately triggers the activation of more executioner caspases. So there is crosstalk between these two pathways. Okay. There are factors that can help the cell survive or block apoptosis. So survival factors will usually lead to activation of transcription factors that trigger the expression of members of the anti-apoptosis members of the BCL family. There are other factors that will cause the inactivation of the BH3 proteins, right? So the survival factor binds to its receptor, triggers and activates a signaling pathway that activates kinases. The kinases can phosphorylate BAD in this case. And when we phosphorylate BAD, this inactivates BAD, and that means that this BCL2 can actually do its job of blocking apoptosis. And there is a third mechanism that involves inactivation of a bunch of different proteins. We're not gonna, I don't really want you to remember that because there's no way to incorporate this in the flow of the lecture. They're not gonna come up on tests or anything. The only thing that you need to know is that this pathway mediated by MAP kinase actually blocks apoptosis, and that's it. Can you folks hear me? Yes, you can. This is working. So now, if you have excessive mitogenic input, that can trigger apoptosis as well, right? So if you have too much of a signal that causes cell proliferation, that can send a signal, like a distress signal that causes apoptosis. So several mitogens bind to receptors that lead to the activation of RAS and of RAS, leading to the activation of MAP kinases. MAP kinase activation triggers the production of MYC, which is this immediate early gene that is a master transcription factor that regulates the expression of a bunch of different things. Excessive production of MYC, because there's remember there's too much mitogen that leads to too much MYC. That leads to the production of ARF and basically the inactivation of MDM2 that stabilizes p53, as we said before, and the levels of p53 can lead to either arresting the cell cycle, or if there is far too much signal to proliferate, then we can actually go into apoptosis. Now, necrosis, I'm just going to say a couple of words about it. The cell death may involve several different things. All of them lead to the loss of cell integrity. The important mediators are enzymes like phospholipase A and lipoxygenases that basically create a lot of radical reactive oxygen species. And that triggers the damage of the cell membrane. And the damage to the cell membrane is sort of the thing that tips the scales towards cell death. Excessive calcium is very, very important here because it triggers pathways that lead to the loss of lysosomal membrane integrity. And if you lose the components of the lysosome, then everything basically goes to hell inside of the cell. Junk kinase, we're going to talk a little bit about junk kinases next week, is also important there. I'm going to actually stop here. What I would like you to basically I think we don't really have a lot more, we don't have that many slides left. One is about necroptosis. There's ferroptosis and peroptosis, and efferocytosis. Those are alternative mechanisms of cell death that kind of look like specialized necrosis, or in some cases specialized apoptosis. I want you to focus for these. I want you to just focus on the definitions. What I would like you to know is what is necroptosis, what is ferroptosis, and what differentiates ferroptosis from pyroptosis, for instance. And the same thing with efferocytosis. All you need to know really is the definitions of each of these processes, and I will actually say just a couple of extra words about autophagy, because autophagy is an important cellular process. Autophagy is recognized as an important process that enables cells to recycle some of its components, particularly components that have, spent enough time. These are oxidized proteins that it enables the recycling of cellular materials. And it does so without triggering cell death. There are basically three forms of autophagy. The more common one is called macroautophagy. And basically this involves the bulk formation, the bulk engulfment of old worn out cellular components like ribosomes, protein aggregates, mitochondria, lipid droplets, etc. The cell basically creates like an envelope of membrane around these old worn out components. And then these enclosed components form what we call an autophagosome. And the autophagosome then fuses with the lysosome, and the lysosome then degrades these components. There are two other mechanisms for autophagy. There's microautophagy and there is chaperone-mediated autophagy. Microautophagy basically involves the direct uptake of proteins by the lysosome. So, there are tiny little worn out components. The lysosome sort of takes them up and then degrades them. And then there is this chaperone-mediated autophagy involves direct interaction of individual proteins with chaperones that then bind to receptors on the membrane of the lysosome and then those proteins are translocated into the lysosome, leading to their degradation. So from here, know the definitions of the different types of autophagy and the general mechanism for macroautophagy, and that's basically all you need to know. I apologize for how long this has been, but we are done. Any questions? Okay, so no questions. I will see you next week with some cell signaling. Thank you for your attention, and I promise it gets better.